Glen, Anheuser-Busch Endowed Professor of Brewing Science at the University of California, Davis, and he will present on his research focused on environmental impacts to barley endosperm quality. Welcome, Dr. Fox.
Thank you very much. Still working. There we go. That's got us going. Thanks. Good morning, everyone. Thanks, Scott, for the invitation to come down. Yeah, I have one of those weird accents, so hang in there. I'm going to talk a little bit about how abiotic stress, particularly heat and drought, impacts quality, but at a much deeper level than just saying, oh, it changes the amount of protein content, which is the go-to measure for industry to say we've got grain that's acceptable for malting grain. We've done a lot of work on starch quality and protein quality to really understand the impact on composition. And thanks to Dr. Bettenhausen in introducing some information about amino acids and already presenting a table of quality data, and Dr. Jin for introducing the concept of composition and molecular weight and how that impacts quality. So when we're dealing with our trial samples or even commercial samples, we understand we might have had a hot finish to the spring and the farmers have suffered a little bit because, you know, it's been a hot spring, so the yield is down, grain size is down, test weights are down, but they still might make malt quality. They might just scrape in at 12.5% protein. And that's how we measure protein. The relationship between protein and the other components within the grain is very important. So we have this sort of natural relationship between protein and starch. High protein usually means lower starch and vice versa. High protein will usually mean higher levels of beta-glucan and arabinoxylan as well. But we don't measure any of those things. We just assume because we've got good protein content, everything else will fall into place. And usually that does happen, but not always. Now, don't be scared. This is just a malt certificate of analysis. A few maltsters have seen many of these and had many conversations with their brewer clients about what a malt COA looks like and the numbers on a malt COA. But it might seem like there's some missing information from the COA that the brewers are looking for. And there might be some information on the COA that's actually important for the brewers, but they may not necessarily see how it relates to their process. But some of the things I'm going to talk about, they're either deeply hidden in some of the numbers of the COA or they're not even on the COA. And I'm not sort of saying we need to introduce more on the COA, God forbid. We don't want to put more on the COA. But there's definitely some stuff missing. If we start with starch, it's just we talk about starch and it's almost like it's a single entity. We talk about total starch, but there's two polymers, and Dr. Jin's already talked about polymers. So these are polymers just made up of glucose. Starch is just a glucose polymer. And we have two types, amylose and amylopectin. Amylopectin is like a tree. There's lots of small branches, whereas amylose is more like just a straight chain. And that's the rough proportions of each. It's around, the textbooks will say 75% amylopectin and 25% amylose. They vary significantly, usually with growing environment, and there are some single mutations where you can actually have 100% amylopectin, or you can have things around 50 or 60% amylose by changing the sequence enzymes. So that's a rather beautiful image, certainly not mine. I stole that one from someone in nature. But the point I want to make is this whole metabolic pathway, and we can see all these things in little colourful circles, starch branching enzymes, starch synthase 1, etc., are all enzymes, and all these enzymes are sensitive to temperature. So while these enzymes are in the plant, and as the plant is making starch, that's all happening. But when it comes to grain filling, and that's when it's getting to the hot time of the year, all these enzymes are sensitive to extreme temperatures. So this is when we can see enzyme performance being diminished. And we see a change in the structure of amylose and amylopectin. We might see a change in the length, the average length of amylose molecules. So as I said, the amylose is basically a straight chain. and the amylopectin is a branch polymer like this. It forms these layers, the crystalline amorphous layers, and then we end up with these lovely starch granules. And we have two basic sizes. They talk about a bimodal distribution, but often the bimodal distribution, there's a crossover in the tails. So we're going to have small, large granules and large, small granules. And the amount of amylose and amylopectin in each of those granules varies. And most small granules have more amylose. And amylose is our problematic polymer. I love saying that, problematic polymers. The other thing here we see these lovely, this is an isolated large starch granule, but the image next to it is got an LS on it. And you can see this small material sticking to the large granules. So that's a combination of small granules and our storage protein. So this is where the granules, the starch and the protein are physically connected. And this has an impact on the efficiency of malting and how easily we can modify our grains based on the proportion or the amount of this hordein fraction. Now, a wee while ago, I measured a bunch of barleys and malts. It was around 300, and we can just see the numbers there. We used to, and still go by amylose content or amylopectin content or starch content. You can just see the extremes at the top and the bottom, 30% amylose and 30% amylose. The x-axis is the average chain length of those amyloses. So you can see there's a five-fold difference in average amylose chain length. That's environment. That's what environment does. It shortens these amylose chains up. It can actually put more branches on the amylopectin. The plant has to do what the plant has to do to think it needs to survive. So it changes all these metabolic functions just to do what it absolutely must do in abiotic stress and situations. Probably the important thing here, and excuse me using the Celsius temperatures, but I am bilingual. I can do the F and C languages, but I do use the C language most. This is really important for the brewers. This is the temperature in the mash tun and how efficient starch is converted to sugar in the mash tun. Now, most brewers are mashing at a closer temperature to 150 Fahrenheit, around 65 degrees C. Starch will gelatinize really quickly at that temperature. Most of the starch enzymes are fairly active at that temperature. But it's when we see gelatinization temperatures, in this example here, that bottom image, this isn't coming up too often, Scott, sorry. Maybe a dead battery. So that top image there is sort of showing the process as these granules become solubilized at temperature. They swell and you get the polymers dispersing out of the granules. The bottom curve is a standard gelatinization curve. Now, if you can sort of see down on the x-axis there, that's a Celsius temperature. The sort of right-hand tail of that curve is when the last of the small granules are solubilized. And we can see that some of those are at 70 degrees Celsius. Other than alpha amylase, and it's only just active at 70 degrees Celsius, the other starch degrading enzymes have come and gone. They're fully denatured. So this is where we're seeing inefficiency in conversion in the mash tun. And I've worked with some maltsters, and they're basically now redesigning with their brewers their mashing protocols because they can actually see these temperatures much higher, and they have to redesign the temperature profile they do with their mashing. And the brewers are actually making, or they're losing less money when they follow these modified processes. And what we're seeing here is, this isn't just a unique thing to North America. My colleagues in Europe are seeing exactly the same things, So it's definitely this sort of shift in, I'm not allowed to say climate change, apparently. So extreme environments. I think it's probably even a better descriptor. So the next area I want to cover is protein content and actually protein composition. And we're all very familiar with protein. Keldale started this. He was working at the Carlsberg lab back in the 1800s. And then Dumas came up with his method. So most people will use a Dumas combustion method to measure protein content. That top image is me on the right doing Keldale's way back in the early 90s. It was a great system. You'd have boiling sulfuric acid right about here. and you'd have boiling caustic right about here. I really don't know why people were never really badly injured, because in those days, and my colleague and friend Ruth Dilmack is here, she worked at the same building as us, and that's a time when people still smoked in laboratories. So I'm really amazed that nobody got badly injured in those days. But the bottom image on the right is the Dumas method, where we can actually do a single seed and measure protein content with a single seed. So going from content, we went to composition, and we moved into electrophoresis. One-dimensional electrophoresis is the bottom left. Then we could do two-dimensional electrophoresis, where we could separate on size and charge. And now we actually have proteomics, which I think is a wonderful technology. Now, people would have seen these sorts of things. We see these lovely fields of barley, and we might sort of take in that barley crop, and the farmer has a sample that's 12% barley. Great, it's malting grade barley. But is every seed 12%? No, it's not. We see this normal variation. So we have some low protein seeds and some high protein seeds. And with that, they built yield maps, and you can build protein maps. And as a consequence, if we've got protein variation, that means we've got starch variation. When we talk about protein composition, and this is really just an absolute minimal representation of the variation in composition, while there seems to be more proteins in the metabolic and protective region, which are the sort of standardized albumins and globulins, the storage proteins are the more important ones. These are the ones that are bound up in the matrix around the granules, and particularly the hordein ones over on the right there. And we talk about those being our main drivers of protein quality. All the others are really minor proteins, whereas those hordeins will make up more than half of total protein because they're in the endosperm. Now, this metabolic map basically shows how all amino acids are formed. Did we get that? We're not. No, it's okay, Scott. We got a pointer?
Yeah.
That one?
No. Well, we just use the mouse. So there you go.
So we can see here individual amino acids. So it goes through all these metabolic processes to produce the amino acids that will then be picked up and made, proteins will be made from those. It's very complicated. Now, all of these also have enzymes in these pathways. And guess what happens to these enzymes when they're under stress? They don't work terribly well. So again, we see changes in enzyme efficiency, which translates to variation in composition. So is this what the protein matrix looks like? And I love this particular image. I wish I could claim credit for that, but I can't. So we see this large granule here. We see some small granules here. And that's the sort of variation in size. And then we see this lovely web-type structure. Well, this is the protein matrix. And for my students, I'm normally doing protein matrix, matrix movie stuff. Wow, okay. They don't understand it either. But it's not just the hordein itself. Bound up in this matrix is beta amylase. Okay, beta amylase is not synthesized in germination. Beta amylase is already there. It has to be released by another enzyme, a protease. But also in this protein matrix is serpin Z4, which is a foam-positive protein. So again, the modification level is releasing some of these important proteins and enzymes. But the more protein matrix we have, then we have more of these potential enzymes, but it requires more work to break them down during malting. So the work we did, and this is for a former student of mine, Marnie Ramanan, who was actually supported with an AMBER grant a few years ago. If we look at the total alcohol soluble fraction, which is basically the storage protein fraction compared to total protein, there is a nice relationship. And the textbooks will again say there's about 50% total storage protein compared to relative to total protein. 50%. That's okay. We can work with that. But this data would certainly suggest we have samples where it's much lower than 50%. And some extreme samples, I mean, that's a ridiculously high protein content sample anyway. So that was heat stress. But nearly 70% of the protein was hordein, which meant it had a very low starch content you wouldn't have been malting that grain anyway, but just shows the relationship that's what's happening during grain development. Now, if we just take the protein content for malting, we might even see a bigger variation. Here's a lovely protein sample at just on 10%. This has got 4% of storage protein, but another sample at 10% protein has 7% storage protein. So again, this is a sample or two samples, lovely protein content, but big variation in storage protein content, which requires they need to be malted differently. But the maltsters don't know that because we don't have information on the storage protein content. We only have total protein content. Again, just diving through some proteomics, the work that Marnie was working on, we ended up with 3,000 proteins with the proteomics we did. We had 70 samples over 3 seasons, multiple locations. So it was really a sort of small G by E on proteomics. If we just took the top 50 proteins, we can see the changes in intensity to red to blue is how much in terms of abundance changes relative to each other. And of course, in that list on the left there are our main hordein proteins, D-hordein, C-hordein, B-hordein, beta amylase. They're just so abundant. But we can see they very much varies across locations because of the growing environment. If we dive a little deeper and add some gelatinization data to this, so that's that curve we saw before where we have changes in starch and solubilization of starch compared to some of our individual hordein fractions, and these are just abundance numbers, and compared to the gelatinization temperatures, compared to total protein, lots of data. So we just do a simple correlation, and we can see that certainly the hordein fractions themselves, where we're seeing, oh, it's green, must be good. No, what it means is more hordein, then we actually have higher gelatinization temperatures. So it's the storage protein itself that's having a bigger impact on gelatinization, not as big as the variation in starch quality, but protein composition is impacting how quickly these granules swell during malting and during mashing. Okay, they actually have physical limitation. And the maltsters will also talk about texture. A soft barley will malt better than a hard barley. And these proteins are controlling texture, hardness. So a higher endosperm protein content, we end up with an inherently harder barley. It's harder to malt, needs more time, but it also impacts friability and milling potential in the brewery. So again, this is a gap in our understanding of what's really going on. What does this all mean? Oh, hell knows. Other than there's still a lot to do, thank God. But we certainly see that abiotic stress, and particularly heat more so than drought, because drought is often a thing that's coming and the plant can see it's coming, it's running out of water. Whereas heat shock in particular, say 15 days post-anthesis, so we have 3 or 4 days where it's suddenly 100 degrees, can really impact all those enzymes that are trying to do their job. And it changes their efficiency. And suddenly there's less starch, or there's less amylopectin and more amylose. Or suddenly we've got more D-hordein, which is the problematic hordein fraction in our protein. We only measure protein content, which gives us little to no indication of compositional quality. We know that this composition impacts on malting potential. And I know the malting industry for a few years now has talked about how can the industry be more efficient? Can we actually make good quality malt with 3.5 days germination? Or can we actually get away with 1 steep rather than 2 or 3 steeps? So the industry is really looking for some innovation there and how they can save some money, time, water, and energy. But it also impacts our gelatinization. So the grain quality impacts brewhouse efficiency. So that's how we relate the grain the farmers grow impacts brewers downstream. And it has a big impact. And we talk about the amount of free amino nitrogen, which is also very important for brewers. Most of this is pretty much completely driven by environment. Unless you're using some weird genetics or it's got some weird genetic background where you've got a starch mutant or a hordein mutant, most of these genes are highly conserved. So unless you're chasing some allelic variation in some of those standard genes, all of this is pretty much environmentally controlled. What's next? Well, we're continuing our work to try and understand some of these relationships. I think we're still trapped with some of our old methods. We certainly have the work we're trying to do is related to the growers' interests. We don't want to suddenly say, well, we're going to change the protein spec. Well, that impacts farmers and how much nitrogen they might apply, how much fertilizer they might apply, and how does that impact yield? Do we want to introduce new protein segregations based on hordein content? No. It's already hard enough to segregate all the barley we have, but it might be something that craft maltsters can work with because they're working with much smaller loads of grain, and they often have a much smaller clientele of farmers they're working with. Do we look at some of these hordein nulls? Because there's been a number of barley varieties released over the years that are missing one of these hordein fractions. And there's a number of hordeum spontaneum lines are missing some of these hordein fractions, and they're happily growing, easily modified. So there's potential out there in terms of some interesting genetics. I have built an NIR calibration. So now on some of our samples, we get protein and alcohol soluble fraction. So my next set of experiments will be to modify my steeps based on my alcohol soluble protein fraction rather than the total protein. And that might be helpful. And there we go.
All right, thank you, Dr. Fox. Do I have any questions?
Thanks, Glen. One question about you said not all the seeds are the same, and this which is interesting by itself. And then you talked about influence of the environment. When you look on the effect on the variation between the seeds under extreme environment, do you see all those components are having more coefficient of variations? Like there is more variation between the individual samples or individual seeds?
Most of that work, when we're just looking at variation in protein content, we didn't get into individual seed composition at that time. And this was measuring protein on single seeds from a sample. We weren't actually going into the field and harvesting spikes, but that work has been done where they've actually measured the protein content of every seed in a spike, and we see still a small variation within the spike itself. And then that's completely transferred across the field if you have a field that might have a nitrogen gradient, nutrient gradient, a water gradient, those sorts of things. So we see this variation. It's reflected in yield as well. So we know we see this variation. It's normally fairly small variation, but it's just an inherent variation we have to deal. And when all the samples come together, we know that we'll get some variation in performance because we've just got that inherent variation.
And you think there would be any value to reduce this variation under extreme environment?
Certainly for the first instance, farmers want to reduce that variation because they might be losing some yield potential if they've got a field where it's slightly high nitrogen and they're overshooting their protein target and their yield might be down. So farmers certainly want to sort of get their fields more uniform in that context, which would then provide grain that is more uniform.
All right, thank you. Sprint across the hallway.
Get your steps up.
Thank you. So you've mentioned two things that are important in the composition. So is it the amylopectin that has the biggest impact on quality, or is it the composition of the protein? That's the first question I have. And then the second question I have, in wheat, we know which protein impacts hardness the most. It's the pure indolence, and we've created mutants that are missing those. Do you know which protein it is impacts hardness the most in barley?
Sure. So thanks, Jamie, for your first question. Heat stress will result in more amylose being expressed because there's only a single enzyme in that pathway to break amylose, whereas there's at least four enzymes in the pathway for amylopectin. And the heat stress will actually produce more smaller granules. So we have smaller granules with more amylose. In terms of texture, it's the similar sort of protein. It's called a hortoindoline. Instead of a puroindoline in wheat, it's hortoindolines in barley. And I did my PhD on this stuff. And we see allelic variation in those. So we can actually see a variation in texture if you've got the right combination of alleles for the two hortoindoline proteins. So they're also connected to that matrix because they're what's connecting the granule to the protein. So again, that physical relationship between the bound up or the storage protein and those granules is very important. And heat stress and drought stress basically creates a much stronger bonding between these. And so when the maltsters are trying to malt and hydrate, it can be problematic in how easy it is for the maltsters to hydrate the grain uniformly because of this physical relationship.
All right. Do I have any other questions?
Thank you, Glenn. It's going to be a bizarre question, as they usually are for me. But one of the old German practices that a lot of brewers do is decoction mashing. So if the modern day brewer is not employing decoction mashing and is working with these barley varieties or malts that have amylose that has a higher gelatinization temperature than that which they're mashing at, would a decoction mash reduce that starch and make it soluble and fermentable?
Absolutely. So the decoction mash normally starts at a much lower temperature anyway, normally around 125 Fahrenheit, and then they remove a portion, heat it up, return that. That's the natural stepwise increase in mashing. So with that process, if they have a double or triple decoction, you basically, and I love the German brewers, from a couple of hundred years ago, because they worked this out, that those temperatures they got to were actually ideal temperatures for the different enzyme sets needed. So the beta-glucanase and isolanase and the protease is working at much lower temperatures. Then they stepped it up. So they could have a poorly modified malt and still make great beer because the decoction allowed a lot more modification to happen during mashing. Modern day brewing is really about efficiency. A lot of brewers are just mashing in at 150 because they can do a short mash. Half the time they don't even know what the COA says, which is the problem anyway. A lot of brewers are not reading COAs. But they do this high temperature single mash, or high temperature mash, and then they might go from 65 to 75 at the end of the mash. But by then, all the enzymes are denatured, but not all your starch is solubilized. So we end up with a lot more undigested starch in wort going into the fermenters. So they have these other downstream problems. So going back old school, decoction or just a slow ramp temperature mash solved some of those problems.
All right. Any other questions?